Welcome back, everybody, to Geology 109, Evolution of the Earth, for the spring 2021 semester at UW Oshkosh. I hope everybody had a good spring break. This marks the beginning of week 9 for the course, where we are starting Unit 3. Over the next 3 weeks, pretty much from this week until the end of the week of April 12th, we'll be talking about the Paleozoic Era. So this is the beginning of the Phanerozoic Eon, the beginning of the Paleozoic Era. We covered the Precambrian on the exam, and now we're continuing to move our way forward. Now, rather than just jump immediately into the Paleozoic Era, I want to talk a little bit first about the transition from the Precambrian to the Cambrian and what's really going on at the very end of the Precambrian Eon. There's a couple of major developments we're going to talk about for the first part of this week's lecture. First, the development of metazoans. A metazoan is a multicelled organism. And now we do probably have these metazoans at the end of the Precambrian, almost certainly do. But at the beginning of the Paleozoic, what we really mark that boundary with isn't multicellular life, but multicellular life with hard parts, because we do have soft-bodied multicellular organisms at the end of the Precambrian, but we mark the beginning of the Cambrian when we start seeing a hard part fossil record, so things like shells. And then in the middle of the Cambrian period, the very beginning of the Paleozoic era, we have an absolutely fascinating and unique deposit known as the Burgess Shale that we're going to talk about too. So if we go back to the very, very end of the Precambrian, remember, during the Archean, we were building our continents, and during the Proterozoic, we were seeing temperatures kind of stabilize, and we were seeing continents produce broad sediment platforms, and plate tectonics continues moving just as it does today. If we look at the very end of the Precambrian to the beginning of the Paleozoic era, this is what the world is actually looking like, and we have a supercontinent. Now, it's not Pangaea. We're not there yet. Pangaea is going to happen in this unit. This is Rodinia. This is another supercontinent that precedes Pangaea by quite a lot of time. You can see this is what the world looked like in the earliest Cambrian, about 550 million years ago. So this is right at that boundary between the end of the Precambrian and the beginning of the Paleozoic era, the Cambrian period. So if we look around here, just from 1 billion years ago to 550 million years ago, we're going from Rodinia to a new orientation we call Pangaea. You'll see, notice where the South Pole is oriented in both of these images and where the continents are. Laurentia is the name that you're going to be hearing a lot. This is ancestral North America. So what we now call the continent of North America is actually the Laurentian Continent. And notice how from Rodinia to Pangaea, notice where Australia, Antarctica, and India are. They're all still kind of clustered together, and they're sticking around by the South Pole, though that does move north a little bit in Pangaea. And really, in the red outline, we see another one of our supercontinents starting to form, Gondwanaland. So when we get towards producing Pangaea, we're going to be seeing the combination of Laurentia and Gondwanaland, these two sub-supercontinents, as they start together, but the stage is already being set at the very beginning of the Paleozoic era. So let's take a close zoomed in look at the very, very end of the Proterozoic era, the Eon, really, the last stage of the Precambrian from about 2.5 billion years ago to 540 to 550 million years ago. And as you can see, we break up the Proterozoic into 3 subdivisions, the Paleoproterozoic, Mesoproterozoic, and Neoproterozoic, respectively. So what do we know that's going on at this time? Well, we talked about at the beginning of the Proterozoic, so really the beginning of the Paleoproterozoic as well, we see this is the time when we're seeing more and more stromatolites and more banded iron formations, because in the Archean we had that continental accretion happening, building up broader cratons and platforms, giving us broad continental shelves for stromatolites and other photosynthetic organisms to diversify upon. This starts producing more oxygen, so we see more banded iron formations. After that, though, we have a major global ice age, where temperatures drop considerably and we start seeing ice sheets advancing on the continents that we have, and we see eustatic sea levels drop globally. When we come out of this, towards the middle to end of the Paleoproterozoic, this is when our acritarchs appear. These are our free-floating photosynthetic algae. Remember, they don't need a stromatolite to stick to anymore, so we see an even bigger boom in global oxygen production because we now have algae floating in surface waters all around the world after this major global ice age. They diversify throughout the Mesoproterozoic, and then at the beginning of the Neoproterozoic, we have another major global ice age. And we come out of that one with the evidence of the first multicellular life, or at least abundant enough to be producing a fossil record. Now, does anybody notice a trend here? We have stromatolites and banded iron formations, major global ice age. When we come out of that, we have this new kind of evolutionary novelty. And then when that diversifies, we have a major global ice age. We come out of that into multicellular life, another big event. So the ice ages here are serving as kind of an environmental upset for the entire planet. And as we know from studying evolution and looking at natural selection, these are the kind of things that help drive diversification. It helps drive different species towards evolution. Remember, acritarchs are significant because they're also the first organism we know of to reproduce with sexual reproduction rather than asexual. So that is mixing more genes and diversifying even more. So what triggers this development in multicellular life? It's what we call the Snowball Earth hypothesis, and it's really kind of an interesting idea. And to really get into this, we have to look at what's called the Albedo Effect. Imagine like a bright winter day after a whole bunch of snow has fallen outside and it's real sunny out, and you step outside and you look and the snow is reflecting the sunlight and it's blinding. You need your sunglasses. That's because snow and ice are highly reflective. They do absorb some solar radiation, but they reflect a lot back, which is why it's so bright. Now imagine you have a planet with ice advancing from the north and south pole towards the equator. Now, at those high latitudes, north and south respectively, you're going to see a lot of reflectance and some absorption. But as the ice continues to grow towards the lower latitudes, towards the equator, it's now reflecting more than it's absorbing, and you get this runaway ice age. And we have evidence that this has happened a few times in Earth's history, what we call a snowball Earth, where you essentially have ice from pole to pole, possibly even covering the equator. Now, this doesn't mean that the oceans are freezing all the way down to the ocean depths, but it does mean that we have significant ice cover over a vast majority of the planet. So this is how Snowball Earth works. About 750 million years ago, still within the lattice portion of the Proterozoic, we see ice starting to advance. And you can see in figure 1 on this image, you can see the ice caps are starting to freeze over. And by 2, they've advanced considerably more. And by 3, this is that albedo effect, the runaway glaciation, because we're now reflecting more light than we are absorbing, so the planet's losing heat. And by the time we get to 3 and 4, we are in true snowball Earth. And the thing is, after snowball Earths, we tend to come out of them very quickly. So it's a quick buildup into it and a very quick buildup out of it. And this is just enough to put environmental stress and ecological stress on a lot of the organisms. And it makes some organisms go extinct, and other ones it gives them the opportunity to flourish. It's kind of like shaking the Etch-a-Sketch, if you understand that reference. So the evidence we have for snowball Earth, this is a really cool hypothesis, but how do we know? Why do we think it happened? Well, if we had ice cover all the way down by the equator at one point in the Proterozoic, we would expect to see glacial deposits at tropical latitudes from the Neoproterozoic, and we actually do. We also see open water glacial deposits, meaning that ice was over the oceans and then dropping glacial debris into that ocean water. So the only way you can really get this is by having this, what we call the snowball Earth situation. So about 750 million years ago, the world froze essentially. It lasts for a few million years. And when we come out of it, we go abruptly into a greenhouse condition where we now have very, very warm temperatures, which is great if you're a marine organism because they usually like warmer, a lot of them like warmer temperatures. So you freeze over a bunch of things and it puts stress on some organisms. They're no longer doing as well, but it gives other organisms an opportunity to do well. And we come out of this, they flourish. So multicellular life pretty much explodes with diversity following the thaw of Snowball Earth. And the reason for this is renewed niche space. An ecological role for an organism is its niche. And once you take something out, that's prime real estate for something else. So every now and then, whether it's a mass extinction from an asteroid impact or a huge buildup of carbon dioxide or Snowball Earth, these are the types of things that happen to our planet that have severe impacts on life, sometimes good and sometimes not. So as we come out of this global ice age, at the end of the Precambrian, we have our first Metazoans, or multicellular life forms. And this is about 700 million years ago, and we refer to this as the Vendian time. It's a little subdivision of the Neoproterozoic. And the organisms we find in the Vendian are referred to as the Ediacaran Fauna. They're all soft-bodied organisms, so think worms and jellyfish. And most of the fossils we have from the Ediacaran fauna are impressions in a very, very fine sandstone or siltstone. If you have anything more coarse-grained, like a conglomerate or a breccia, you're not going to preserve the fine detail that you need for these types of organisms. And the reason we call it the Ediacaran fauna is, even though they have been found in other places, the first location that these fossils were found were in the Ediacara Hills of Australia. So they've kind of been given the type locality name collectively all around the world. So if you're looking at 700 million year old Vendian metazoans, you're looking at Ediacaran fauna. And yeah, as I said, they are found everywhere where we have the right facies of the right time worldwide. So what some of them look like, as you can see, that we're looking at flatworms and jellyfish. So not maybe the most exciting things in the world, but when you consider that it's the first multicellular organisms, that is pretty exciting. Also note that they're small, but they're not tiny. I mean, this flatworm, for example, is at least about 4 inches wide, which is pretty good sized. What's really cool is you can look, especially in the upper right, that impression in the upper right is actually a jellyfish, and they don't look much different than the types of impressions you would get from a modern day jellyfish today, if it were to land in sediment. These organisms haven't changed considerably in all of this time because they're very, very simple organisms. Now they're complex for being multicellular compared to single cellular, but compared to some of the other things that we've seen, they are a little bit simple. So our modern soft-bodied marine invertebrate animals, not that different. We still have polychaete worms and flatworms and jellyfish alive today. And if you think about it, this requires a very special kind of fossilization and preservation because they don't have hard parts. So you need that soft impression. And without lagerstatin like this, without that exquisite preservation of soft tissue being represented, we wouldn't even know about an Ediacaran fauna. And this is really where we start drawing the boundary, or shortly after this, for the beginning of the Paleozoic Era, starting with the Cambrian period. Okay. About 550 million years ago, we have our first fossils, representations of our first organisms that show the ability to make shells or mineralized hard parts. Now let's think about what this requires. This means you are ingesting certain materials and your body is growing a new tissue that is made of something mineralized. So it's not just cells making simple cells, it's cells using calcium, using carbonate, or carbon and oxygen, or in some cases phosphates, to make hard parts in or outside of the body. And there are some great advantages of this. First of all, you can have really, really strong muscles, but it really helps if you have something hard to anchor them onto. So hard parts give organisms a substrate to anchor on movable parts and their muscles to. And obviously, shells are armor. It's great protection from predators, which also means we now have predators at this time as well. So there's this nice kind of evolutionary arms race going on where you have organisms that are going to develop hard parts to keep themselves from being eaten. And that's going to then push for a drive for the carnivores to develop ways of getting around that armor system. And it keeps growing today. that keeps developing. So we're going to be focusing on the Paleozoic era here. I just want to highlight this for a second. We're going to be looking at this section for this exam. Okay, so after the Vendian, before the Mesozoic era, we're looking at the Paleozoic era. Notice this is the first beginning of the Phanerozoic Eon, which starts with the Paleozoic era, which of course starts with the Cambrian period. So the Cambrian spans from about 550 to 488 million years, give or take a few million years. It kind of depends. One book might say 555, one might say 550. We're all kind of in that same ballpark though. So here's some of the earliest Cambrian fossils. We do still have acritarchs at this time. And we have what we call our Timoshan fauna, this small shell fauna. Some of these we don't even really know what they are. This is a hyolithid here on the right, and we're not even 100% sure what a hyolithid is. Is it a type of mollusk? Is it a type of brachiopod or something, or maybe a relative of a coral? We're not really sure. It looks to be mostly mollusk-like, so something like a cephalopod or maybe a gastropod, something like that. Other ones are a little more easier to identify. H, for example, is pretty obviously a mollusk. We look at B and C, we're not really sure. E could be a small part to something much larger, but the important thing here is these are biogenically grown, so organisms are growing these mineralized structures, and we call these earliest Cambrian fossils the Timoshan fauna. Now today, most invertebrate organisms, like that you'd see leaving shells on a beach, are making those shells out of calcium carbonate. So chemically, it's the same stuff as calcite. In the Timoshan fauna, we're looking mostly at phosphatic organisms. So they weren't really making a lot of calcium carbonate shells yet, but they were making their shells out of calcium phosphate. Around the middle of the Cambrian, kind of early middle, we have what's called the Cambrian Explosion. And this goes from a few dozen genera to nearly 700 in only about 30 million years. Now that may seem like a long time, but remember, so far in this course, we've been dealing with things in terms of billions of years. So after the Timoshan fauna, which is where we get our first shelled organisms, towards the end of that early and towards the middle of the Cambrian, we have the Cambrian Explosion. And notice the way that life is just diversifying here. We start out with just a handful of organisms, and now here we are with that many. And it dips up and down a little bit, but that's the first huge spike of life that we get in the Cambrian. Notice here's our Vendian, just to keep ourselves oriented. Yep, that's where we're still looking at mostly those soft-tissued organisms. Timoshan fauna, we have our first shelled organisms here in the early, and then by the time we get into the latter portion of the early Cambrian, that's when we're seeing this huge spike in diversification. Let's take a look at some of the organisms we would see in the Cambrian. If you wanted to go to a reef in the Cambrian period, and reefs are very important. We're going to be talking about reefs a lot because they are a major source of biodiversity. If you wanted to go to a reef in the Cambrian, this is as good as you'd get. It would be an archaeocyathon reef. So they're not corals. They're more like sponges, but they're also a little bit different. Notice in the cutaway view here, you have kind of a double-walled basket with these partitions in between, and then all of these pores. Now, modern-day sponges do have tiny little fibers that will move back and forth, and it'll help pull water in and through their baskets to help them filter feed and suspension feed. We don't think archaeocyathons had that. They probably just sat there and waited for water to flow through them, and when it went into those little vestibules, those little pockets in between their double-walled cones, that would decrease the water velocity, and organic material would fall out of suspension onto the cells, and they could digest them. Now, this probably doesn't sound like they'd be getting a lot to eat, and you'd be right. But when you're not getting up and moving around or really doing anything, you don't need a lot of calories or nutrients. So this is a very effective way of existing. Now, You can see they're relatively small. There's a penny there for scale next to a cross-section of an archaeocyathon. So they wouldn't be very big reefs, but we do know that they came in a wide variety of shapes and sizes and different orientations. One of the most popular types of fossils from the early Paleozoic, especially the Cambrian, are the Trilobites. Now we're going to be tracing Trilobites for the next couple of weeks because they're a really good marker for changes that are going on in the Paleozoic era. We start out the Paleozoic in the Cambrian with Trilobites, which are a type of arthropod, so they're related to, distantly related to insects and spiders and shrimp and lobsters and crabs and so on. But we have a lot of different trilobites in the Cambrian. In fact, their diversity, the numbers of trilobites, is really, really high. However, just looking at the screen here, they all look pretty similar. I mean, there are some subtle differences. These two down here, we're actually not 100% sure they are trilobites. Either they're the most primitive trilobites or they're trilobites that are either the most primitive trilobites or the closest thing to being a trilobite without actually being one. But the rest of them all look pretty similar. This would mean that their disparity, degrees of differences, is low, but their diversity, number of species, is high. So we have a lot of species, but they all look alike. So high diversity, low disparity. And this means they're all probably doing something very similar. They're all filling very similar ecological niches because they're not diverse or disparate enough, different looking enough to indicate a wide variety of functional morphology. Now, the word trilobite means 3 lobes. And this can get a little confusing because we have 2 different ways of describing the lobes of a trilobite. The head structure of a trilobite is known as the cephalon. Now remember, you're looking down on the back of the trilobite, so it's actually the back of its head. This entire big structure here, this big boomerang-shaped thing, is the cephalon. The main body portion of the trilobite is the thorax, and then you have the tail portion, which is the pygidium. Now, trilobite, 3 lobes. These are not the lobes you're looking for. No, the lobes you're looking for are the axial lobe, which is the center of both the cephalon, thorax, and pygidium, and then the left and right pleural lobes, respectively. Okay, so this actually works as kind of a coordinate system. We can talk about how the axial lobe of the pygidium on one trilobite is different than another, or how the left pleural lobe of the cephalon is different on one trilobite than another. It kind of gives you an XY coordinate system to use. And this is really one of the ways that we kind of use to describe the differences in trilobites. What's going on with the cephalon, thorax, and pygidium at different times. Let's talk a bit about the Burgess Shale. The Burgess Shale is one of the most famous and important fossil localities in the world. It represents the Middle Cambrian, so about 505 million years old. And again, it's been found in a number of different places, but we're going to be talking about the type locality. Okay. We know a lot about the Cambrian ecosystem because of the Burgess Shale. And here you can see a photo of it in the Canadian Rockies, and where people are walking. It's a very thin shale deposit, hence the name. It's very, very thin, paper-thin mudstones that's incredibly rich with Cambrian fossils. The Burgess Show was discovered by Charles Walcott in 1909. Interesting true story. This was a trip that Walcott and his wife took on the back of pack mules for their honeymoon. He convinced his wife to go with him on a fossil hunting expedition. So that was pretty cool. And they discovered this now world famous fossil locality. So this is Walcott. He was actually the director of the U.S. Geological Survey throughout the late early 1900s. Very important paleontologist and also a member of the Smithsonian Museum. Yep. So a paleontologist, secretary of the Smithsonian. So, Burgess Shale, Middle Cambrian. What's so great about this? We get more than just hard parts preserved. In fact, the hard parts preserved here are pretty poor quality, but we get really, really good preservation of soft tissue. So, organisms that died and were buried in what is now the Burgess Shale, their hard parts didn't preserve as well as the soft parts did, which is really strange. But we do have both. which means we can get more information than just looking at a shell. We can see how the shell attached to the entire body of an organism. And this has revealed an absolutely incredible amount of organisms, different diversity, many different species, including some things that we have no idea how to classify them. And that's something that's kind of the take-home message of learning about the Burgess Shale is extinctions have happened in the past. There are organisms that have existed in the past that we can still see relatives of today. And then there's things that went extinct in the past that's it. There is no living group surviving that extinction event from that. So this is a unique, what we call a rare window into the Cambrian, so into life from the past during a time when we had a lot of different radiation of life, including new kinds of predators. So this is when our ecosystems start to become incredibly more complex, and we have this cool paleontological window into it. And because we have so many bizarre organisms, we've nicknamed those hopeful monsters. These are things that worked really well for this particular environment, this particular time and space, but a little bit of change in the environment and they just don't work anymore. They just don't fit. It's being too specialized for your job. So here's some of our Burgess Shale fossils. Now these images look a little grainy because they're taken with polarizing light. So you can see some of the soft tissues, because normally if you just look at the fossils with the naked eye, it looks like a black shale with this weird shiny thing on it. But when you look at it through different light filters, you can see all sorts of incredible structures. One thing I'd like to point out though is this guy down here. This is Pikia. Notice the parallel structures going down the body of Pikia and this line that runs down its back. You are looking at the earliest chordate. We are chordates. Fish are chordates. All vertebrates are chordates. Now this is not a vertebrate. It doesn't have bones yet, but it does have a flexible notochord, and those parallel lines going down really looks a lot like a fillet of fish, because those are called myomeres. That's how fish grow their muscles still today. So this is a small, just a few inches long, jawless fish ancestor. Our ancestor from the Burgess Shale, from the Middle Cambrian. So this is one of the earliest relatives of what will eventually be fish and then other, later tetrapods and vertebrates. But then we have all this other weird stuff. Things like Opabenia and Anomalocaris, like these guys. This one is really strange. It's called Hallucigenia. It's a worm-like organism that has like a pincher thing on one end. It's got tube feet and spikes, but we don't know if it walked on the spikes or walked on the tube feet. It's a really, really strange organism. So we have segmented worms. We have our early chordates. And then who knows what Hallucigenia is. Opabenia might be an extinct phylum, meaning we don't really know how to classify this weird proboscis-bearing, multi-lobed, multi-eyed predator. So it could just be an extinct phylum. Hyolithids, which we talked a little bit about at the beginning of this lecture. Now we have more of the full body of them, and they're still really weird. Maybe they're a type of mollusk, maybe an extinct class of mollusks. We don't know. Anomalocaris could be an extinct phylum or an early arthropod. One of the cool things about this is what you're looking at here is a scanning electron microscope image of its mouth. Those are all of its tooth-like structures. So yeah, it had this horrible hole of sharp teeth and those little feelers on the front of its mouth to shove food into that nightmare. And then yeah, down at the bottom there's Pikia again, this little kind of stalk-eyed fish-like organism. These are really, really rare though. I do want to mention that Pikia is a very, very rare chordate in the Burgess Shale. You don't find them very often, and that's really important, and we'll come back to why that's important here in a little bit. Again, Hallucigenia, and I love the name for that because you'd have to be hallucinating to come up with that thing. Extinct phylum, we have no idea what that is. And then we do have some stuff that we're pretty sure of, like Canadapsis perfecta is pretty clearly a crustacean. It's a crab-like organism. It's very similar to like a brine shrimp today. So some of these things haven't changed much in 505 million years, and some have changed a lot, and some have gone extinct, and we have no idea where to classify these. So what is the Burgess Shale, though? Or what kind of deposit are we looking at? So notice the scale bar here. That's 30 centimeters. These are very, very thin layers of shale. So layers of these muddy sediments are positioned adjacent to a big algal limestone cliff underwater. So imagine an underwater cliff. And obviously it's a cliff, so the top of that cliff is going to be more in a shallow water environment. But if you go over the edge into the deeper water, that's where you're going to be building up all of this mud, all of this muddy sediment. So you have organisms living on top of that algal reef platform in the shallow waters, and then storms come by and they shove a bunch of them over the edge. So those sediments and some of the organisms rain down off the edge of that cliff into deeper water and are deposited as the Burgess Shale. Organisms are going to be buried or smothered, and this is important too because it's a deeper water environment. There's less oxygen. They're buried quickly. It's exactly what you want if you want a preserved soft tissue preservation. So here's like a cross-section of what the Burgess Shale would have looked like. The organisms would have been living up here on that thin, in what is now the thin portion of the Stephen Formation, which is what the Burgess Shale is in. And a storm would come by and down over the edge they'd go, and they would get buried in the various deposits of the thicker portion of that Stephen Formation. So they're probably living on top of what we're calling the Cathedral Formation, which is this algal limestone reef. And they go over the edge, and they're buried in essentially an anoxic basin, or a deposit with very little oxygen in the bottom waters. Now, when that happens, in really, really deep environments, sometimes calcium carbonate can start to re-dissolve into seawater. So that's probably one of the things that's happening is they get shoved over the edge, deep water, the carbonate starts to break down a bit, but there's not a lot of oxygen for decomposition to take place, so we get these really nice preservations of both the hard parts and the soft parts. So here's kind of a three-dimensional view of what this would have looked like. Get rid of all these to help us out visualize that a little bit more. So the organisms would have been living on top of that platform and getting shoved over the edge back in the Cambrian and being buried. And as you can see, the Walcott Quarry is what's annotated in the red dot, and all those yellow dots are other places in the Canadian Rockies where we have Burgess Shale type deposits. So following the Cathedral Formation, that escarpment, we can actually find more Burgess Shale type localities. So here's one of our classic paleo reconstructions of the Burgess Shale, and I love images like this, whether it's Burgess Shale or late Paleozoic or dinosaurs, because it's like the family picture where everybody gets into the photo when in real life things aren't nearly this congested. But you have a little bit of everything represented. You've got polychaete worms, you've got Chenidapsis, Anomalocaris, hyolithids, Opabenia, and you've, of course, got Hallucigenia and Pikia and little Oaxia running around, all of these different type of mid-Cambrian organisms. But let's talk a little bit about the importance of the Burgess Shale, because it doesn't just represent a Middle Cambrian fauna, which is important. It lets us know what was going on in the Mid-Cambrian, and the preservation is important, but there's more that can be teased out if we think about this in a big picture of what this means about the evolution of ecosystems on planet Earth. This is the first evidence we have of a complex ecological community. Think about it. Before this, it's pretty much stromatolites, which is a colony of single-celled organisms stuck to sediment photosynthesizing. That's cool, but now we actually have more things happening. We have benthic and pelagic organisms. Benthic means you're living in the bottom of the sea. Pelagic means you're living in the water column. You're free swimming or free floating. So we have things that lived at the bottom, that walked around, and then we have things that were swimming. We have filter feeders. We have organisms that are pulling water through their bodies and filtering out organic material to eat. We have organisms that are burrowing down into sediment, utilizing the space and the nutrients that are encased in those muddy sediments as well. We have predators that get up and move around and chase after their prey or ambush their prey, but they don't sit in one place forever. They can get up and move to new hunting grounds. Now, when Walcott recognized that we have all of this incredible diversity of life in the Burgess Shale, he went through and tried classifying all of the organisms that we saw in the Burgess Shale into our modern day classifications of animal phyla. So he looked at Anomalocaris and said, it looks like an arthropod, put it in with arthropods. Or Hyolithids, looks like a mollusk, put it in with mollusks. And in some cases, yeah, that makes sense. But there were some other ones that didn't really work, but they kind of got shoved into those proverbial boxes anyway. We now actually have gone back and reviewed much of the Burgess Shale fauna and now see that while we do have some modern phyla represented and classes represented, there's a number of organisms that probably went extinct, and that was it. There is no more phyla, whatever. That would be the equivalent of if all of phylum Arthropoda went extinct. You would have no more bugs, no more spiders and scorpions, but you'd also have no more crabs, lobsters, shrimp, butterflies, anything like that would be gone. So by today's standards, we can say, oh, wow, if we got rid of arthropods, that would actually be a vast majority of life on the planet. If we got rid of all echinoderms, that would be no more sea stars, no more sea urchins and sand dollars. We're talking about that level of extinction that perhaps early on in the mid-Cambrian, we had organisms that were really, really unique, and they don't fit in any modern day group of organisms because they're entirely extinct. And that brings us back to Pikia, our little chordate, our little vertebrate ancestor from the mid-Cambrian. And remember we said this little critter, which is a distant relative to all vertebrate life, including ourselves, is very, very rare in the mid-Cambrian. Now, the reason that that is important is because a paleontologist named Stephen J. Gould wrote about the Burgess Shale quite extensively in a very, very good and very accessible book, Wonderful Life. And Gould brought up an interesting point. First, I want to point out that Stephen J. Gould is the only paleontologist that has ever been featured on The Simpsons, where he did his own voice work. But what Gould proposed was what's called the contingency of evolution, saying we know that plenty of organisms in the Burgess Shale went extinct, and that was the last line of a major classification of life. Chordates were not a very important or diverse phylum in the Cambrian. We have Pikia and a handful of others, but we don't have a lot for chordates. So they're kind of like hallucigenia in that way. They're just another weirdo, another hopeful monster in the Cambrian. If you could go back and rewind the clock to the Cambrian period, and whatever made things like hallucigenia or Anomalocaris go extinct, if it, for whatever reason, would have taken out Pikia, this little rare fish-like thing in the Cambrian, we would not be here. So from an unassuming origin, just from random chance, it happened to make it through whatever was going on that caused these other things to go extinct at the end of the Cambrian. Pikia and its relatives, though rare and not significant portions of the ecosystem, perhaps they were slightly better generalists, made it through. And 550 million years later, here we are. Of course, there's a lot of stuff that happens in between, and that's what we're going to be talking about for the rest of the semester. So the Precambrian Cambrian transition, just to review real quick before we continue on. We begin this transition talking about the development of Metazoans. Remember, this starts at the very end of the Precambrian, and they really get a kind of a boost because of the Snowball Earth mix-up, right? That causes some ecological stress, which puts pressure on some groups of organisms and gives others a leg up. So we have our first Metazoans, first multicellular life with the Ediacaran Fauna, the Vendian period, very, very end of the Precambrian, about 700 million years ago. The dawn of the Paleozoic is marked by the Timoshan fauna, the early Cambrian hard part fossils. And then we see the diversification of Cambrian fossils with the Cambrian Explosion. Rapid diversification of life about 505 million years ago. So 700 million for the Ediacaran fauna, About 550 million for the Timocian and 505 million for the Cambrian Explosion. And we talked about the Burgess Shale, looking at the importance of finding soft tissue morphology in Cambrian life. It allows us to get a better idea of what these organisms are. We start seeing the first complex ecological communities and the significance of unassuming rare species having a huge impact later on down the line. And that brings us into the actual Paleozoic era. So let's start looking at the Paleozoic, specifically at the Cambrian and the Ordovician periods of the Paleozoic era, and focus on the environments and the type of living things we have. We've talked a little bit about some of the early Cambrian life, but I want to look at more what makes up this early Paleozoic fauna. Also with that, we want to look at what kind of tectonic and evolutionary changes are happening in the early Paleozoic, what kind of evolutionary innovations we're seeing with some of these organisms, and the continued complexity of ecosystems that we first see the origins of in the Cambrian. So let's start just with tectonics and environments of the early Paleozoic. So I know we already looked at this earlier in this lecture. I just want to remind you once again, we're looking here at the Cambrian period, 550 to about 488 million years ago. But what we're going to be talking about for the rest of this lecture is the Cambrian and the Ordovician. Next week, we'll look at Silurian and Devonian, and then the third week of this unit, we'll look at the Carboniferous and the Permian, which brings us then to exam 3. Now, we know that over time, sea levels have gone up and down around the planet. When they go up, you have a better chance of depositing sediment, and when they're down, you erode sediment. So what we have here are what we call our megasequences. In other words, these are time periods when we have significant transgressions and then regressions throughout the North American craton. So what you're looking at here is a diagram that shows the deposition of sediment across what is now North America over the last 600 million years. But it's just looking at major sequences. So to kind of point this out, the dark green that you see here and here and here and so on, those are times and spaces where we don't have sediment deposited. In other words, it's an unconformity. So if you go back to before 600 million years ago and you look at the North American craton, we don't have much for sediment at that point in time. But what we start noticing is throughout the Cambrian, the late Vendian, we have a little bit of a transgression that continues up through the Cambrian. And by the time we get to the end of the Cambrian, beginning of the Ordovician, you'll notice how we have sea levels, or we have sediments that go all the way across the North American craton. That means sea levels were high. And then we drop them back down in the Ordovician, and they start to go up again. So every time you see a sequence go all the way across, whether it's Salk, Tippecanoe, Kaskaskia, Absaroka, Zuni, those are major global transgressions. So that's what we're seeing here is transgressions bounded by unconformities. The unconformities are representing the regressions. So we can say then that during the Cambrian, whether you're looking at the east coast or west coast of North America, there is a sea level rise. Sea levels are going up during the Cambrian, and we call this particular deposit of transgressive sediments the Salk sequence. So that represents an encroaching sea level going up higher and higher and higher and higher until it hits a maximum in the early Ordovician, and then it drops, and we start a new sequence in the Ordovician called the Tippecanoe, and we'll get to that in a few. So the Salk transgression is a sea. It's the Salk Sea. This was the first time we had in the Phanerozoic a global transgression flooding the North American or Laurentian continent. And anytime you have a continent that is underwater, we call it an epieric sea or a continental sea. It's essentially a continent being flooded. And during the Cambrian, we see evidence of ongoing transgressions across Laurentia. Now, we happen to be positioned here in a part of Wisconsin where the Salk Sea is present in its sedimentary form. This is where the Salk Sea was. And in fact, if you look at North America, this is looking specifically at Cambrian sediments in North America during the Cambrian, the Upper Cambrian, so the Late Cambrian. Notice we have everywhere that's white kind of in here, going all the way down to this transcontinental arch. That would have been essentially the continent. Everything else is platform. So we have sand piling up around the transcontinental arch, and then in the deeper water we're getting limestones from the Cambrian, and then far out we have these deep water shales. Everything else south of that didn't exist yet. There was no Mexico, there is no Florida, there is no Appalachians. All of that's not even present yet. Texas doesn't exist yet. All of that's gone. It's not even there. So the sand that we have piling up just around that transcontinental arch is eroding off of the craton where I put the big X. And it gets blown around. Now, there's no plants on land yet. There's no animals on land yet. You just have rock eroding into the sea. Now, this is showing you what's called an Isopack Map. We already looked at what types of sediments from the Cambrian we have in North America. Let's look at how thick they are. And this is in meters. And if you look very carefully right at the shield to transcontinental arch, it's 0 because that's where you have crustal rocks. But as far as sediment packages, we get out to some areas here where those shales are 3,000 meters thick. So these are very, very, very deep and thick packages of Cambrian sediment. Now we can take this one step further and actually look at what kind of sediments we have here in Wisconsin. We have a lot of this Cambrian sand in Wisconsin, and hopefully the name Salk sounded familiar because a lot of this is in Salk County, which is actually where we get the name the Salk megasequence. So today, a lot of the sandstones that we mine out of Wisconsin that go to foundries in Neenah, Wisconsin, and various other places, and are used for 3D printing or making glass or making steel products and iron products. A lot of the sand that they use for that comes right out of here in Wisconsin, and it's all Cambrian in age. In fact, if you look at a bedrock map of Wisconsin, all of this tan stuff is Cambrian sand. This portion is all Precambrian part of the shield. So when we talk about the transcontinental arch and the sand piling up right up next to it, that's what you're looking at. The bright colored stuff that I outlined there in the north, that's all part of the actual shield. The sand that we have around here, around Wisconsin, that is so abundant, is all Cambrian in age, and it's what was eroding off of that Canadian shield 500 million years ago. And this is what it actually would have looked like. Now, I want to point a few things out. First of all, notice how the equator is oriented. That's because North America, or Laurentia, was actually rotated 90 degrees. So the equator was still 0 degrees latitude. But this is showing you kind of in our modern orientation of North America. Would have actually been straddling the equator. And that craton, or that shield, really would have just been a few sandy islands and rocky islands sticking out of this shallow sea. So during the late Cambrian period, sea levels were at an incredible high, where most of the continental crust we have is underwater. And the little bit that's sticking out is constantly being eroded and hit by wave action, remobilizing these little quartz grains, and they get tossed around over and over and over again. After that transgression and then a regression, we go into the Ordovician period. This is from about 488 to about 440 million years ago. So these are going to be the rocks on top of our Cambrian Salk deposits. If we look at the paleogeography of the early Ordovician, most of our continental masses are assembling, plural, in the southern hemisphere. Laurentia, or Ancestor North America, is still kind of hovering around the equator right next to Siberia and Greenland, and Baltica is in the southern hemisphere. So we still have a long ways to go to get to our modern orientation of continents. Around the mid-Ordovician, so about 480 million years ago, the regression begins, and this is what really drops and ends the Salk sequence. We lower sea level, and a lot of those sands that were deposited in the Cambrian and lower Ordovician become remobilized. And now that they've retumbled and been recycled and redeposited, they're deposited into a new set of facies. We call this the St. Peter Sandstone Formation. The St. Peter Sandstone. This is a huge marine aquifer, but we don't find a lot of fossils in it. In fact, a vast majority of the water that we have in the Midwest is stored in the St. Peter Sandstone. So it extends all the way down to southern Oklahoma, but it's underneath a lot of limestone and dolostone by the time you get there. But here in the upper portion of the Midwest, it's very, very close to the surface. So as it rains and as we get snow melt, it fills up that sandstone unit from the middle Ordovician. And this sand is really quite unique looking too, if I can wax poetic about sand for a moment. It is like sugar. It looks like sugar. It looks like it's this very white sandstone. It's really, really strange. And it's really slippery because the grains have been recycled so much they feel like little ball bearings. So this is used a lot also in making glass. The St. Peter Sandstone is also a good aquifer, and it's also very good for making glass. A lot of our other upper Ordovician facies, though, a lot of our local rocks include carbonates like limestones and dolostones and shales. Again, all things that we would see as the Tippecanoe sequence. Our next major sequence is starting to transgress. So we have the Salk C, which is a transgression. It hits a maximum, and then we drop that with a regression in the middle Ordovician. And then throughout the rest of the Ordovician going into the Silurian, we have the Tippecanoe sequence, our second major rise in Phanerozoic sea levels. Something else, though, is happening in the late Ordovician. Not only are sea levels going up with our Tippecanoe sequence, but in what is now our east coast of North America, we have a trench, a subduction zone. And as we are subducting plates under what is now New England, back in the Ordovician, we start uplifting a significant portion out of the sea and producing the first uplift event for what will be the Appalachian Mountains, and we call this the tectonic orogeny. Now, when we think about the Appalachians, they're the smaller mountain range in North America. We think about the mountains of North America, that's the Rockies, the big Rockies out west. And we'll get there. The Rockies are relatively recent. The Appalachians are very, very old. And it wasn't just one uplift event. It's actually a series of a bunch of uplift events. The first of which is in the Ordovician, and we call it the tectonic mountain building event, or the tectonic orogeny. And I want to point out that at one point, and we'll get there, the Appalachians were the size of what the Himalayas are today. Now today, you can hike over the Appalachians because they've been eroded down for hundreds of millions of years. But we're going to actually be tracing the series of impact and uplift events that caused the Appalachians to grow to the might that they once had. And this all starts really in the Ordovician with the uplift of the tectonic highlands. Now what's going to happen as we uplift this land is we now have rocks sticking up out of the sea, and anytime they stick up, they get eroded down. So now we start spreading out sediment into this what's called an alluvial plain. Now remember, this would be north of the Appalachians because of where the equator is and how things are positioned, but by today's standards, these are what we find westernmost of the Appalachians. All of those sediments that were uplifted with the tectonic origin or mountain are now shed down towards the Midwest. So we're here in the Tippecanoe sequence. We raise sea level in the Cambrian, dropped at the beginning of the Ordovician, the middle of Ordovician. Then we start building it back up again. And notice that we have a longer period of time where our sequence is all the way across the craton. Our sea level goes all the way across. With the Salk C, it was a relatively short-lived high stand of our transgression, but the Tippecanoe lasts considerably longer. So the Ordovician and Silurian and Cambrian, really mostly Cambrian Ordovician, these are time periods of generally high sea levels. Now let's talk a little bit about what kind of organisms are living in this changing time from the Cambrian and the Ordovician. So this is kind of how we'll be going through a lot of our geologic periods, is what's going on geologically and tectonically, what does the stage look like, and then we can talk about what the players are doing on that stage in terms of life and ecosystems, and in this case, evolutionary innovation.
As we move through the Paleozoic, though, and actually even onward into our modern times, I want to introduce you to the concept of different marine faunas. Now, when we're going to look at these three, the Cambrian, the Middle Paleozoic, or just Paleozoic fauna, and the modern fauna, we're talking about organisms being replaced, but not entirely. What we're really looking at is what are the dominant groups of marine organisms in these different periods of time. So for the Cambrian, going into the early Ordovician, we have what's called the Cambrian fauna. And this means that the dominant organisms in the oceans at the time were trilobites, what we call inarticulate brachiopods, which are a type of filter-feeding organism, very primitive or archaic mollusks, so early types of clams and snails, and very primitive crinoids, which are distant relatives of things like sea stars. Now, in the Middle Paleozoic fauna, or what we just call the Paleozoic fauna, we still have some trilobites and inarticulate brachiopods and all those other things, but we have more elaborate and more diverse organisms as well. We have different types of those brachiopods. We have sea stars and bigger crinoids. We have corals, but different than the ones today, and more developed mollusks. And then in the modern fauna, which really starts at the beginning of the Mesozoic era, this is when we get our modern groups of bivalves and snails and shellfish like crustaceans, and then all of our bony fish, which again bony fish go back to the Ordovician and Silurian, but this is when they become really dominant marine organisms. So I don't want you to think about these different faunas as completely 100% replacing each other. It's just more replacing who's in charge. Again, if we look at the trend throughout the Ordovician of our trilobites, remember we mentioned that trilobites were very abundant in the Cambrian. We had lots and lots of different types of these arthropods in the oceans, but they all looked very similar. Well, we lose a lot of them at the end of the Cambrian. We lose even more at the end of the Ordovician. But in the Ordovician, the trilobites we have are fewer in numbers. We don't have as many different species, but the species we do have are much more elaborate and distinguishable from one another. This is a trend we see throughout the rest of the Paleozoic, that trilobites become more and more rare, but the ones that we do have, the species are more distinct. So for trilobites throughout the entire Paleozoic, they become less diverse and more disparate, meaning that we have fewer numbers of species, but they become more distinct. A lot of the distinctions that we see from them are innovations that help them protect them from predation. For example, even looking at the trilobites that we have here, you can see that this particular type, which we call Cryptolithus, has this kind of lacy brim around its cephalon. That's because it, along with these really kind of flat shovel-shaped trilobites, would burrow under the sediment to hide from predators. Other ones, like the phaecopid trilobites, the Clemenes and phaecops, would roll up into a little ball to protect themselves from predators. So a lot of the innovations that we see in any invertebrate organism are to, find ways of staying alive when there are more and more predators. Here are some of the other extremes that we can see trilobites doing, from having eyes up on stalks to being covered in spines, the ability to burrow in sediment or enroll into a ball. All of these are factors that are features we consider that are used to help ward off predators, or at least make it harder to be eaten by predators. I also want to take a moment to just shout out to fossil preparators, people that handle fossils strictly with the job of restoring them and cleaning off sediment from them. The trilobites that you see here and here, especially with those very delicate needle-like spines, were painstakingly prepared and restored, sometimes scraping away little grains of sediment one at a time to preserve these things. So when you go to a museum and you look at really, really nice fossils, keep in mind that somebody's job is to prepare those, and it's almost an art skill in and of itself. Well, one of the reasons why we have all these diverse trilobites or disparate trilobites and all of these spikes in armor is because we have new predators in the Ordovician. Predators that still have some ancestors in our oceans today. The dominant predators, though, in the Ordovician were cephalopods. Now, cephalopods today are mostly represented in the form of squid and octopi, but we also have cuttlefish and nautiloids, and the nautiloids are the ones that actually we see as the top predator of the Ordovician. So if you think about a squid or an octopus, they don't have any hard parts really. They don't have a shell, but the nautiloids do. So these were gigantic squid-like organisms that had this chambered shell. Each shell was broken up into little compartments, like here and here and here and here and here. Each one they could fill with water or pump water out of. Now, if they fill all their chambers with water, they're heavy and they sink to the bottom. If they pump water out of those chambers, they become buoyant and they can rise up. And if they do it quickly, they can propel themselves through the water like a bullet. So cephalopods in the form of nautiloids had these chambered shells, which we now call an orthicone. And some of these things got really, really big. For example, just to kind of point this out to you, the phragmocone or orthicone can be 10 centimeters to 4 meters in length. So you're looking at a shell that could be around 12 feet long. And those are what were the dominant predators, for example, here in Wisconsin in the Ordovician. So if you went swimming in the Tippecanoe sequence or sea, these are the types of things that might try to grab you. Today, we still have a chambered nautilus alive. You might see these at aquariums, and if you bisect their shells, again, you can see the open chambers held together with a tube that was called a siphuncle, where you can see the attachments to that. So it allows water communication and air communication all the way through the shell. They have this fleshy hood called an operculum. If they don't want to be bothered, they can pull all their tentacles inside of the shell, close the door, and they can just hover in the water depending on how much, how filled their chambers are. So today, these are the only living cephalopods that have an external shell. We have a couple of different types of nautilus today, the chambered nautilus, the paper nautilus. So two genre, I think 3 or 4 species, but back in the early Paleozoic, that's pretty much what we had for cephalopods, were these shelled, chambered type nautiloids. And today we just only have a few different species left. Most of our cephalopods today are represented by squid or octopus, which is a totally separate group of cephalopods called coleoids. Corals also were around in the Ordovician, some of our first corals, and you might have been scuba diving or snorkeling in a coral reef before, and I guarantee the corals you looked at in that reef are not the kinds that we had in the Ordovician. We have 2 major groups of corals in the Ordovician, the Rugos and the Tabulates, and these are all now extinct. The corals that you see alive today are a separate group known as scleractinians. They don't evolve for hundreds of millions of years after these. After the Ordovician. The Rugos corals are solitary or colonial, meaning that the animal, the little polyp of actual animal that makes the coral structure, could either be one per structure or many. And your tabulate corals are all colonial. So there's lots and lots of different tabulae, or little rooms for different polyps. So when you look at a tabulate coral, for example, there's little floors and ceilings. Each one of these would house a polyp or an individual animal, and they have these little windows that they would extend their little tentacled, stinging celled arms out of and wait for something to come by so they could zap it, pull it in, and eat it. On our solitary Rugos corals, each coral structure would have a single polyp living in it like that. So the Rugos corals have what are called radiating septa from the center. This gives the polyp something to grip to as it's growing, and this is actually looking at a colonial version of a Rugos coral. Okay, so Rugos corals would grow in these colonies or as solitary. Sometimes the solitary ones are called horn corals, and the Rugos colonial ones, such as this specimen here, which is called hexagonaria, is a very common kind of a honeycomb-looking coral. Your tabulates, though, all colonial. And we've got 2 major ones here. There's Halosites, which is here, sometimes called the chain coral, and then Favosites, which is what we're seeing more represented here. Again, each one of these housing thousands of individual organisms that secrete this calcium carbonate structure to produce these big reef frameworks. And just like modern day corals, we think these things probably did live in relatively shallow waters, because modern day corals have an algae that lives inside their cells that photosynthesizes and thus feeds them. So it's called zooxanthellae. It lives inside their bodies. It's a symbiotic relationship that will allow the coral to get some of its nutrients from these algae that live within them. That's why corals today have to live in shallow water. It's because they have a cell inside them that photosynthesizes. The polyp itself doesn't, but it gets its nutrients from that. So if you take a coral and you put it in the dark, you're slowly starving it, because it's one of its main nutrient sources is from this type of algae. We also have a group of organisms in the Ordovician and onward called conodonts. Now, conodonts are weird because their fossils look like these weird little spiky plates. And these are actually very important for the petroleum industry, because these are made of calcium phosphate. They're made of the same things that your like bones and teeth are. And if they are found in sediment that's near very high-grade petroleum, they will look jet black. If they're near lower grades of petroleum, they can look kind of brown. So some paleontologists are employed by oil companies to look at sediment cores and identify conodonts and get an idea of what kind of grade petroleum is nearby. Conodonts are actually, though, a type of fish. That's what a conodont animal actually looks like. They had these gigantic eyes, and those spiky plates are their mouth parts. Now, they look like jaws and teeth, but those aren't actually. They couldn't open or close their mouth. Their mouth was permanently open, and those spiky structures were essentially a net that was inside their mouths that would help filter out all of these different types of microorganisms. So they'd swim around and just filter feed or suspension feed, and they're very small worm-like fish. So that's what a couple of different conodonts would look like in different perspectives. They look kind of like minnows, but they all look permanently surprised. They have big eyes and their mouths are permanently stuck open because they don't have a lower jaw. Jaws don't evolve for quite a while in fish. However, bones in some forms of jawless fish do evolve in the Ordovician. These are the ostracoderms. Most of their skeletons are still cartilaginous. Think about kind of like sharks. But ostracoderms were covered in bony plates, so they were kind of a flat fish with no jaw. These things probably ate algae off the seafloor, so kind of like a Roomba, but with fins. And they had all this bony armor all over their bodies, probably to keep themselves safe from big orthocone cephalopods. So we've talked about jawless fish and conodonts. We've talked about carnivorous nautiloids, a little bit about other kinds of gastropods and cephalopods. We have our scavengers like trilobites, and then sponges and corals and all these other things. This is when we start seeing even more increased complexity in our ecosystems in the Ordovician, where we have a true food web starting to be formed. So we hadn't seen this type of complexity. We started to see complexity in the Cambrian, but it gets even more so in the Ordovician. And there's another group I want to mention, and that's the echinoderms. So echinoderms today are represented by sea stars and sand dollars and sea urchins and sea cucumbers and things. But in the Paleozoic, the most common type of echinoderm were these things called crinoids. And they look like plants. In fact, their nickname is the sea lily, but this is actually an animal. They're really, really strange. Echinoderms, the name means spiny skin, and that's because their bodies are covered in a leathery kind of coating, and under that is a skeleton that is made of calcium carbonate crystals. So they're made of all these plates, but each plate is a single calcium carbonate crystal that are grown. They don't really have blood and tissues the way we would think of most animals. They do everything through hydraulics. So they pull seawater into their bodies, and they will pump seawater from one part of the body to the next to make it move. What looks like roots down here at the bottom, those are actually tube feet, but again, made of a series of calcium carbonate plates. The stalk is made of a series, if you could look at this up close, it's little tiny disc-like rings, all stacked up on top of each other. and they can move. These arms and pinnules, they look like needles, but they actually are all hydraulic moving structures to grab organic debris floating through seawater and passing it down to the arboreal cup, which is where the mouth of that echinoderm is. Now, when these things die and their fleshy bag covering them decomposes, the plates just kind of scatter. So a lot of times you'll find rocks made entirely of echinoderm parts. And that's because once they die, they disarticulate very quickly. But there's something else that these steampunk animals suggest that's very, very important for our understanding of the evolution of ecosystems, and that's what we call tiering. If we look at our Cambrian faunas, we notice that everything is pretty close to the sediment-water interface. Everything's really close to the seafloor. We have some things that are burrowing down a little bit, and some things that are stalked up a little bit, but not much. But in the Ordovician, we start seeing our crinoids grow taller, and some of our bivalves grow deeper. And that's because they're utilizing space above. And this is exactly what we see in other environments like rainforests. In a rainforest, you have organisms that live in the leaf litter and in the ground. You have things that live in the tree trunks themselves, things that live in the upper canopy, the lower canopy, and so on. Now imagine a coral reef or any kind of marine environment. You have organisms that live deep underwater in the sediment, things that live on the sediment, and things that grow up high above the sediment in the water column. The Ordovician is the onset of what we call epifaunal tiering. This is when we start to see organisms not just growing side by side to other species, but up and above. So to kind of conclude our early Paleozoic, we looked at the Cambrian and the Ordovician, and this is from your textbook. Notice that we have three things we're looking at on this diagram. We have what's going on with our biotic events, our geologic events, and then sea level. Throughout the Cambrian, we see the continued transgression of the Salk sequence. While that's happening, we're still moving some continents around, but as sea levels are going up, we start to see the diversification of life in the Cambrian with the Cambrian Explosion, the Burgess Shale, and then we start seeing some trilobite extinctions towards the end of the Cambrian. And as sea levels get very, very high in the Ordovician, we see continued diversification of our Paleozoic style faunas, as we talked about here. We have our jawless fish, different kinds of brachiopods, which we'll talk more about in the Silurian. But we then end the Salk sequence with a regression. And then as we're building up the Tippecanoe sequence, again, we also have the tectonic orogeny happening. We end the Ordovician with a major glaciation in the southern hemisphere and a mass extinction. So the thing I really want to point out is this relationship right here between sea level and the diversification of all of these marine organisms. We see this continued diversification of the Paleozoic fauna, new kinds of predators and jawless fish when sea levels are at an all-time high. That's the kind of relationships we're looking at. Once you have higher sea levels, there's more places and more types of niches for different types of marine organisms to utilize. So we'll go ahead and pause here, and next week we will be looking at the Middle Paleozoic. And in lab this week, we'll be looking at more on Paleozoic life. Thanks.